of LMSK, feel welcome to this webinar today. Today's webinar, we are taken through this presentation by Mr. Eric Rono, and this is very important because of our place in the medical sector. Some few more announcements before we start. One, this webinar is KMLTTB accredited and will attract 3 CPD points. Secondly, according to the KMLTTB regulations concerning the provision of CPD points, the medical laboratory practitioners are required to attend a minimum of 60% of the webinar's duration to be eligible for the CPD points. So once again, feel welcome. And my name is George Okoth Simbo, and I'm privileged to take you through this session as your moderator. And we are also privileged to be taken through this session by Mr. Eric Kipkwech Rono as the presenter, and today's presentation is on flow cytometry. So just some snippets of the presenter's introduction. Our presenter today, as I've mentioned, is Mr. Eric Kipkwech Rono. Mr. Eric Kipkwech Rono is a seasoned clinical laboratory scientist. He's also a laboratory systems consultant and a PhD candidate in microbiology. He is specializing in microbial diagnostics and host pathogen interactions. So rest assured that Mr. Eric Kipkwech Rono brings over 20 years of experience in medical laboratory science, including leadership roles in diagnostics, clinical trials, and advanced biomedical research. Mr. Rono previously served as a laboratory manager at the Kombewa Clinical Research Center, which is the Cambridge Water Reed project. This is in Kisumu. And in that capacity, he led both clinical and research operations with a strong focus on quality assurance, the CAP, ISO compliance, and team capacity building. Mr. Rono has a deep expertise in flow cytometry. He has served as deputy laboratory manager and section head at Cambridge Water Reed in Kericho. And his work has included immunophenotyping, cell activation profiling, and biopsy analysis. And this supporting major HIV and immunology research efforts across East Africa. And at this juncture, allow me to invite our presenter, Mr. Eric Rono. If you can hear me, kindly take the stage and proceed. Thank you very much to this far. Karibu sana.
Thank you, Mr. Osimbo, George. As you have heard, my name is Eric Rono. Currently, I consult for the Ministry of Agriculture. I also consult for the Africa Clinical Trials Solutions and also the Mahabara Africa. I'm based in Kisumu. That is where I'm presenting this from. Our topic today is advanced application of flow cytometry in both clinical and research diagnostics. This is an area where in Africa we have never done so much, but when you travel to other countries,
you will really know that flow cytometry is an area that Africa needs to venture into. Just the rationale of why we are doing this today is because flow cytometry is a powerful analytical technique used to detect and measure physical and chemical characteristics of a population of cells or particles. It is not only cells that flow cytometry can do, but also other particles that can be tested. With advances in monoclonal antibodies, for those who have been in the field of flow cytometry, you are aware about the monoclonal antibodies. You're also aware about the fluorochromes and the software analysis that are employed in flow cytometry. Its applications have extended beyond hematology into immunology, oncology, infectious disease, and vaccine development. There's a need, as I said before, to enhance understanding among scientists and healthcare professionals to fully utilize this technology in diagnosis of diseases. The main objective of this presentation today, by the end of this presentation, participants will understand the basic principles and components of flow cytometry, identify key applications in clinical and research diagnostics, appreciate the relevance of flow cytometry in immunophenotyping, disease monitoring, and cell sorting. And then eventually we'll discuss the current trends and future innovations in flow cytometry. The basic principle of flow cytometry is it is used to analyze the physical and chemical characteristics of cells or particles, as I said before, as they flow in a fluid stream through a beam of light. Typically, a laser, we can have the red laser, we can have the blue laser, we can have the argon laser, and several of them, depending on the on what you really want to study. We also have what is known as light scattering. It measures the cell size. It also measures the granularity. For the people who have been in flow cytometry, you really know what is FSC, that measures the size, and also the SSC, that measures the granularity, or what is popularly known as internal complexity of the cell. In addition, there are monoclonal antibodies that are conjugated to the antigenic sites of the cells, and that brings us to the fluorescence detection. Uses fluorescently labeled antibodies or dyes to detect specific molecules. When the laser excites these fluorochromes, they emit a light at characteristic wavelengths, and the cells are detected. The key components of a flow cytometry, we have what is known as the fluidic system, where it aligns cells in a single file within the sheet fluid so that they pass through the laser one at a time. The optics also come into play, where we have the lasers that provide a focused light source to excite fluorochromes. There's the collection lenses that captures scattered and emitted light, and the optical filters that discriminate what kind of light that needs to pass, or what kind of light that needs to proceed based on the wavelength for different detectors. Then we have the detectors themselves and the electronics, where we have the photodiodes
for the scatter and for the photomultiplier tubes, popularly known as the PMTs, for the fluorochromes. And then converts lights into electronic data, and then data is acquired and analyzed. Digital signals are processed by specialized software, which produces histograms and dot plots for interpretation. Some of the outputs is the scatter plots, the dot plots. They differentiate the population based on their sizes, granularity, and fluorescence, which we have talked about. And also, it comes in forms of histograms. It shows fluorescence intensity or other parameters for the population. Key applications of flow cytometry. We have in clinical diagnostics, where we do immunophenotyping. For example, the T cells, B cells, and natural killer cells. The monoclonal antibodies are used to stain the cells, probably the blood sample, and then classifying and quantifying immune cells populations. Critical for diagnosis of leukemia. We know we have several leukemias that are currently available, ravaging our country. We have the ALL, we have the CML, we have the several leukemias that are happening. We also have the lymphomas and the immunodeficiencies. Flow cytometry also is used for disease monitoring, tracking residual diseases after treatment, monitoring immune statuses, like for those people who are HIV positive. The levels of CD4 and CD8 counts are monitored over time. It also in cell cycle analysis by detecting abnormal proliferation in cancer diagnostics. In research applications, we have the cell sorting, isolating specific cell population for downstream analysis. Functional studies, analyzing cytokine production, apoptosis, and cell activation markers. For the gentlemen and ladies who are in immunology, you are aware about immunophenotyping, and then you are aware about also intracellular cytokine staining, where cytokines are stained that will go deep inside the cell. The cell wall are broken, and the dye is taken straight into the cell, and staining done, and analysis done in flow cytometry. We also have cell activation markers like the CD38 markers. We have HLA-DR markers that a study can be done based on what you intend to find out in a cell. It is also used for vaccine development, monitoring immune responses at cellular level. This is done mostly in pharma, where vaccines are being produced. Monitoring immune responses at cellular level is the way to go. We also have the stem cell research, characterizing and isolating stem cell populations. We also employ this flow cytometry in infectious disease and also microbiology. For example, in bacterial detection, identifying and quantifying pathogens using flow cytometry, mostly done in other countries. I'm not aware in Kenya where they do this. I know a few places that they do flow cytometry in Kenya. I know Kemri, I know Kilifi, I know Kericho, I know CDC also does flow cytometry. I know at some point Kisian were doing under CDC, they were doing schistosomiasis by flow cytometry. That is an area that they had explored under parasitic detection. We also have the antimicrobial susceptibility testing, measuring viability after antibiotic exposure. I wanted to bring out just an example of how to diagnose ALL. This is acute lymphoblastic leukemia diagnosis. Just to give example of how it is done. We know that sample collection for this is by bone marrow aspiration or peripheral blood sample can be used. A sample is obtained via bone marrow aspiration, which is an exercise that has to be done by an expert, or the peripheral blood, depending on the suspected diagnostics. For ALL, a bone marrow sample is often preferred because it gives better results. So sample preparation for this is a sample is processed and stained with a panel of fluorescently labeled monoclonal antibodies that targets specific cell surface markers. The sample is then passed through the flow cytometer, where individual cells are analyzed by one based on their sizes. We know the forward scatter of a flow cytometer gives us a size. Like if you are doing immunophenotyping, we have the monocytes, we have the lymphocytes, the lymphocyte being the smallest of the cells, and then followed by the monocytes having a medium size, and then the granulocytes, the neutrophils, the eosinophils, and the rest. So that there is a way that the flow cytometry dot plot will look like. Those cells that have the smallest size will stay on the lower side, having a lower those that have a small size will have a lower forward scatter. Those with medium will be at the middle, and then the granulocytes will be on the other side of the forward scatter. The sites for the side scatter for the fluorescence also, you know the different kinds of fluorescent antibodies dyes that can be used. For example, the FITC, the PE, the PACP, the APC, and so forth and so forth. There are several dyes that can be used. So selection of antibodies and markers for the ALL diagnosis. For diagnosis of ALL, the key is identifying the lymphoblast population and confirming their lineage, whether they are B cell or T cell, as well as their stage of differentiation. The antibodies chosen will target specific cell surface markers and help distinguish these cells from normal hematopoietic and other forms of leukemia. Selection of antibodies and markers. We have the B cells, as I have said. For example, we have a CD19 marker on this kind of B cells. A pan-B cell marker, typically expressed on B cells at various stages of differentiation. You also have a CD10 marker, normally known as the color. Early B cell markers, often expressed on blast in B cell ALL. CD34, a marker, a stem stroke progenitor cell marker, indicating an immature stage of differentiation. CD22, a B cell marker expressed on more mature B cells and their progenitors. And then we have the TDT, that is more specific. It's called the terminal deoxynucleotidyl transferase, a marker for very this is a marker for very early BRIB and T cells. Its presence in ALL plus confirms immaturity. Then we have the T cell acute lymphoblastic leukemia markers, where the CD3 there, because being this T cell, they have a CD3 marker. It is a T cell marker present on the surface of all T cells, all T lymphocytes, or all T cells. We have the marker cluster of differential 7, another T cell marker often present in T cell acute lymphoblastic leukemia. CD34, as mentioned earlier, it marks immature cells and is present in both T cell and B cell precursor stages. TDT also present on T cell blast in T cell ALL. There are general markers for leukemia that I thought I should also mention. The CD45 marker, common in all leukocytes antigen, whether it's a B lymphocyte, whether it is a T lymphocyte or a T helper. So CD45 is a marker for all leukocytes or the white blood cells. It helps to distinguish leukocytes from non-leukocytes, like the red blood cells and platelets. Those doesn't have the CD45 marker. So it helps us to distinguish when we see our dot plots. I normally say our colors and our flowers in flow cytometry dot plots. And then leukemic plast typically express CD45 dimly or in an abnormal pattern. We also have the human leukocyte antigen, which is expressed on some hematopoietic cells, especially the blast cells. Flow cytometry data collection. The flow cytometer detects cells that bind to the fluorescent antibodies. The data is then presented as a dot plots, histogram, or a density plots. These are two different fashions of how the cells appear or presentation of this data. It can be dot plots, it can be histograms, or density plots. For example, the dot plots, typically the forward scatter versus the side scatter is used to differentiate cells based on the size and granularity. I've said that several times, the side scatter and the forward scatter. Lymphoblast in ALL usually have a low forward scatter and a low side scatter. So in immunophenotyping, the general word for immunophenotyping is just having to know what is on the antigenic sites that are outside the cell. Somebody needs to really differentiate between immunophenotyping and intracellular cytokine staining. Immunophenotyping is mapping what is outside the cell. Cells are further analyzed by plotting fluorescent intensity for each antibody. In ALL, lymphoblast will be CD34 positive from the dot plots that you will see, or the histogram that you will see. It will also have the TDT markers, it will also be positive for CD10, and either CD19 positive, CD19 positive for B cell ALL, or CD3 positive for T cell ALL. Normal T or B cells will have a different combination of markers and will not exhibit the abnormal pattern seen in leukemia. So interpreting results for this B cell ALL and e.g. precursor B cell ALL, they are positive for CD34, CD10, and CD19 and TDT. Negative for CD3, no T cell markers. And then these results indicate an immature B cell lineage consistent with acute lymphoblastic leukemia. T cell acute lymphoblastic leukemia, positive for CD34 also. You see, it is also positive for CD7 as opposed to CD10 for B cell ALL. It is positive for CD3, and also the same is also positive for TDT, which is also positive in the B cell ALL. So how to differentiate this is because this has a CD7, which the other one doesn't have. And also it is CD3 positive. It is negative for CD19. It is negative for CD19. It doesn't have the CD19 marker, which B cells ALL have. These results indicate an immature T cell lineage consistent with T cell acute lymphoblastic leukemia. The final diagnosis and reporting, flow cytometry report. The report typically includes clonality, the presence of a monoclonal population indicating a clone, immunophenotype, a detailed list of surface markers and their expression patterns, the lineage assignment, whether it is a B or a T cell origin, and then differentiation status, early precursor cells, e.g. CD34 or TDT. And then percentage of blast, a higher percentage of abnormal blast indicates more aggressive disease. Example report for a B cell ALL. Flow cytometry reveals a predominance of CD34, CD19, CD10, and TDT, positive blast, with an immunophenotype profile consistent with precursor B cell lymphoblastic leukemia, B cell ALL. The blasts are negative for CD3, and also they are negative for CD20, confirming the absence of T cell or mature B cell lineage. For example, in packet ALL, CD45, a common leukocyte antigen, it helps to distinguish leukocytes from non-leukocytes. Like red blood cells and platelets, leukemic plast typically express CD45 dimly or in an abnormal pattern. HLA-DR, human leukocyte antigen, which expressed on some of hematopoietic cells, especially the blasts. This is just an example of a dot plots of what I was explaining. You see the green cells down there. These are positive for their site scatter is low, these are lymphocytes that are taken a certain dye. They are CD45 positive. These are plus in ALL can be can be identified using the site scatter versus CD45 plot. These plus have low site scatter, many times smaller than the normal lymphocytes, and dim to negative CD45. This differs from plus in AML. It differs. Sorry. Example of peripheral blood with CD45 negative B lymphoblast and characteristically small, low site scatter. These are just B lymphoblasts that are CD45 negative, and they have a low scatter. Example of a bone marrow with CD45 dim B lymphoblast. You see there we have different kinds of fluorescent dye. It can be a CD19 PE. It can be CD10 PC5. CD34 PC versus Cy79 PE. So depending on what you are looking for, the selection of these monoclonal antibodies and how you would want to do your kitting based on what you are looking for. Like in pre-B ALL cells, they co-express CD10 and CD19. Expression of CD34 and TDT indicates immaturity and is characteristic in pre-B acute lymphoblastic leukemia. CD38 is an expression of an activation marker, showing that there must be something that is going on in the patient's body. The phenotype of the blast is independent prognosis. BALL is subdivided into early pre-BALL. It is TDT positive, CD19 positive, and CD10 negative. Common ALL, it is CD19 positive, CD10 positive, and also color positive. Pre-B ALL, they are CD10 positive, or it can be CD10 positive or negative, but CD19 positive. HLA-DR, they also have sometimes the cytoplasmic IgM positive cells. And then mature B acute lymphoblastic leukemia, they have CD10, they are positive for CD10, positive for CD19, positive for CD20, positive also for CD22. Service markers are also available for IgM for this kind of mature B ALL. So flow diagnosis, if you want to really rule out, is that for BALL, it is CD45 downregulated, CD19 positive, TDTD positive, CD10 positive, and then service light chains are negative. Conclusion. Flow cytometry is the cornerstone in the diagnosis of acute, just as an example, for acute leukemia, providing rapid, accurate, and detailed immunophenotyping analysis of leukemic cells. The choice of antibodies is based on the specific cell lineage and the maturation stage, with B cell ALL and T-cell ALL requiring distinct panel of markers. This diagnostic approach is crucial in confirming leukemia, subclassifying it, and determining the best therapeutic approach for the patient. The future innovations. We are heading to the AI technology. You see even in the social media that there are some jobs that will end in the next few years because of the AI technology. But what I'm very sure is that lab sciences will not end because we will just get things to improve what we do, like integration with artificial intelligence and machine learning, AI-driven algorithms for automated gating in flow cytometry, and many other lab sciences. And pattern recognition will reduce analysis time and improve accuracy. We also have what is known as a microfluidic flow cytometry. These are miniaturized platforms for point-of-care diagnostics and low-cost applications. You will realize that the flow cytometers are very expensive. The fax counter itself is to the tunes of millions and millions of dollars. The fax caliber, the Fortesa, the Aquos flow cytometry, they are very expensive equipments. But I know BD and Peckman Coulter are working on microfluidic flow cytometry equipments, small equipments that can be used at the level of the dispensary or at the level of an hospital that is in the village. We have also single-cell multi-omics, combining flow cytometry with genomics, transcriptomics, and proteomics for a holistic view of a cell biology. We will also have a label-free flow cytometry, whereby using techniques like Raman spectroscopy and holographic imaging to characterize cells without fluorescent labels. You will realize that when you do flow cytometry without proper compensation or setting up of the equipment or being trained, you will really have erratic results because of a spectral overlap of the different dyes that are used in flow cytometry. For myself, I was trained in Germany, a place called Heidelberg, the Center of Excellence for Flow Cytometry under BD. I've also trained in South Africa under a National Institute of Communicable Diseases, where I did my intracellular cytokine staining. So for this flow cytometry, I would hatch the different sectors in our country to train the personnel adequately, so that they will be able to face the challenges that comes with the technology of flow cytometry. We'll also have portable flow cytometers. We'll also have the portable flow cytometers that battery-powered devices for field diagnostics, especially for infectious diseases in resource-limited settings. You will realize that the current flow cytometers are bigger. If you know the LSR, Life Science Research flow cytometer, it is as big as it's the biggest, one of the biggest equipments that we know under flow cytometry. If you know also of the fax caliber, it's also huge. If you know of the fax canto, it's bigger than the fax caliber. The Fortesa from BD, I know our course is a smaller equipment from Beckman Coulter. And I know we are drifting towards the smaller equipments that are portable and also that can use battery for diagnostics. Thank you very much for listening to me. That is the much I had for you today. I know in the future also we will venture into the nitty-gritty of diagnostics. I just used an example of how we can diagnose ALL, but we can also in future have as a webinar on intracellular cytokine staining, where we can really zoom on the cytokines, the different kinds of cytokines, like the IL-2, IL-6, and IL-10, and several cytokines that can be detected by flow cytometry. Thank you. I welcome questions. Thank you so much.
Thank you very much, Mr. Eric. Indeed, it's a wonderful presentation, and we really appreciate you being there and also presenting for us on this platform. So at this point, there are several questions that I hear. I'll start by Trevor Jim. Trevor Jim is asking, what other common surface markers can be characterized by flow cytometry other than CD? What other common surface markers can be characterized by flow cytometry other than CD? CD, the complementary differentiation. Over to you, Eric.
Yeah, so thank you, Jim. That's a tough one. I'm very sure. I'm very sure that there are other markers that can be looked at apart from the CD markers. I know we have the CD markers are so many, but I'm not very sure about that unless somebody who is an expert also in flow cytometry can chip in on this. I need to check any other marker that can be done.
Okay, thank you very much for that response. Justin Getonto is also asking, what advice will you give on live? What advice will you give on live or dead discrimination when optimizing your assay on ALL? This is acute lymphoblastic leukemia. What advice will you give on live or dead discrimination when optimizing your assay on ALL?
Yeah, so long as you have used the right monoclonal antibody and you have set your equipment very well, yeah, yes, and you have done the compensation very well, I don't think you will go wrong with your results. Sorry. Sorry.
Thank you very
There's a
much.
flu that is going around Kenya that is really so tough.
Okay, I hope Justin have got that clearly. Peter Ngeno. Peter Ngeno is asking, explain the role of MHC molecules in T cells activation. Explain the role of major histocompatibility complex molecules in T cell activation and contrast this with how B cells recognize antigens. Yeah, is a two in one question. you will explain the role of MHC molecules in T cell activation and contrast that with how B cells recognize antigens. And then it's going ahead to say, include the significance of antigen processing pathways. Yes, over to you.
George, thank you for that question. Yeah, so we know MHC class 1 molecules are very crucial part of the immune system, responsible for presenting antigens on the surface of cells. That is antigens on the surface of cells to cytotoxic T cells, triggering an immune response against infected or cancerous cells. So these molecules are found on almost all nucleated cells, and proteins are essential to distinguish self from non-self. That was a tough question from Ngeno. But if you can, you can check. We have the structure and the location. MHE class molecules are composed of heavy chains and beta chains. And in humans, the main class 1 molecules are HLA, LHLA-B, and LHLA. So their functions is to present peptide fragments derived from proteins to the T cells. Those are CD8 cells. And also, when cells are infected or become cancerous, it may produce abnormal or foreign proteins, which are then processed into peptides and presented by MHC class 1 molecules. It needs time to explain that. Probably we can have another CBD on that and explain further on the same. Thank you.
Okay, thank you very much for that. Very many of our participants are appreciating this presentation. Faith David is saying great presentation. I really appreciating. Yeah, Bartolomeo Imbuka is really appreciating and is saying good elaboration on flow cytometry and many others as we proceed. So Edwin Kipkorir has a question here. What are the key pre-analytical and analytical variables that affect flow cytometry results in routine lab diagnostics? What are the key pre-analytical and analytical variables that affect flow cytometry results in routine lab diagnostics? Yeah, so Edwin is concerned with the factors that affect flow cytometry results as you are going about it routinely in the lab, and is focusing on pre-analytical and also analytical variable. But you can also talk about post-analytical. So just respond to it in a wholesome manner. So over to you, Mr. Rono.
Yeah, so if you are doing a blood sample and you know you need to lyse the cells, the red blood cells. So if you don't do it correctly, using the correct ratio of, you dilute the lysing solution, it might impact on your results. Two, during the staining, you must also use the correct monoclonal antibody titer. I know some say 10 microliters and some say 20 microliters, but you must do the validations to actually know what kind of titer are you going to use for staining your cells. It is very important to do a validation study before the beginning of any study, and actually validate that using this method will qualify you to have good results. Also, the equipment setups are very important. Setting up the equipment. If you are using cells that have been like the PBMCs, the PBMCs have to be quality PBMCs. They have not been exposed to they have not been exposed to temperatures that will affect their viability. Other things that need to be watched is storage of your reagents. You must store your reagents in the right conditions so that the reagents that are using are quality, and also they are not deteriorated. I know some places do use expired reagents. Where I worked before, the use of expired reagents was not allowable. But I know in the government facilities, they say probably they are still working. But before you use that, kindly validate with another reagent that is non-expired for the same. The other thing is on analytical issues. You should have an expertise on an assay that you are doing. Train if you have not been trained adequately, say it loudly, because this trial and error can really give us things that probably, if somebody else is given to interpret, we are in the wrong direction. So be talking, and you should be shouting the loudest if training has not been done to you by the organization where you are, and also consult widely to other experts in the field of flow cytometry. Thank you. I know there are other things, so many of them, that can be done for quality results. If you can actually check on AI or ChatGPT, it will give you quite a number of things that you need to observe when doing flow cytometry. Thank you.
Great, wonderful. Thank you very much. There's another question. Martin Juguna is asking this question. Is it possible for cytochemistry to differentiate blast cells? I think he's saying about flow cytometry. Yeah, and attaching it with blast cell. Is it able to differentiate blast cell? Over
It
to you.
depends. Yeah, it depends on the dye that you're using. You see, as I said before, what you are looking for, you have to choose the best methodology. So if cytochemistry,
Yeah, just a minute, Mr. Rono.
yeah,
Martin Juguna is putting more emphasis, more clarity, saying that he means blast cell morphology. Yeah, so I think it should read that. Is it possible for the cytochemistry or flow cytometry to differentiate blast cell morphology? Yes, over to you.
I think yes. I think yes. So you see, as I said before, flow cytometry, you use the dyes, Cytochemistry also you use specific dyes. So my answer for now is yes, I think it can differentiate.
Great. So yeah, Lydia Jeptom is appreciating the presentation, and she's saying great presentation. Yeah, Roslyn is also appreciating. Patrick Osango is appreciating the presentation, and many others. And here we have David Chege is concerned with the cost. What is the cost of the basic tumor markers at your facility or any other facility, or maybe in Kenya or any other place? Yes, what is the cost of the basic tumor markers? Over to you, Eric.
Wow, for the cost, I cannot really say I know exactly how much it is. Because for me, I've been in research. Probably somebody who is in Agagan who does flow cytometry could be good knowing the cost.
Wonderful. So I think this is a variable that keep changing. So I think it can be sought. So David Chege, yeah, you can check on, other places just as be directed by the presenter on the cost. And here Roman Wambua is having this concern. In the current diagnostic practices in Kenya, can flow cytometry be used as a conducive test for diagnosis of ALL, considering the other histological and hematological methods? Can flow cytometry be used as a conclusive test for diagnosis of ALL, keeping in mind that there are other histological and hematological methods currently in Kenya? Over to you.
Yes, I can say flow cytometry is more precise, more accurate than any other methodology. It has been proven. I've seen in other places, there are other equipments that can be able to do a lot of multi-parametric analysis. What I can say is flow cytometry is accurate and more precise. And probably in future, as we move, probably the country can employ, especially the Kenyan government can venture into this area because it is more precise and accurate. In other countries where I've been, in the US, flow cytometry is really mostly used in the diagnosis of ALL. Also in South Africa, they use flow cytometry for ALL diagnostics. Also, I've been to Bangkok, Thailand, and that is where they really have the fancy flow cytometers that they use for the diagnosis of ALL. Thank you.
Yeah, this is wonderful. And Evelyn is also joining me in saying wonderful presentation. So Evelyn Webala is appreciating your presentation. How to differentiate hematogones from the blood cells on bone marrow samples of a pediatric patients? So Ochieng Oroa is asking, how can one differentiate hematogones? Hematogones are the normal immature B lymphocyte precursors that are found in bone marrow. So for those normal but immature B lymphocyte precursors in the bone marrow, how can they be differentiated from the blasts in a pediatric patient? Over to you, Mr.
Yeah,
Eric.
my question also to Ochieng is, How has he been doing it? Can you share a bit of how he has been doing it so that probably we can improve on what he has been doing?
Okay, that's wonderful. Ochieng or Roa, you can just lift your hand. You just cut up your hand, and then you can just explain a bit on what the presenter has just asked. Yes, so you're allowed to talk. You can just unmute and talk, Mr. Ochieng Orua, and just give an oral format of your question so that we can respond accordingly, or rather the presenter can respond to you accordingly. Ochieng Orua, if you can hear me, you can just unmute and give a verbal question or response. Once again and finally, Ochieng Orua, if you can hear me, you can unmute so that your question can be well attended to. It seems Ochieng Orua is not uh, close to his microphone. And by that, we'll just move to the next question. Emmanuel Ngugi is really appreciating you, Mr. Eric, that indeed this is a good presentation, and is affirming that flow cytometry is very useful in this era of HIV management, among other diseases. So thank you very much, Emmanuel Ngugi. Among other, there are quite a lot of appreciations that are coming in here. Yeah, Chengoro is saying that his microphone is not working at the moment, and he has just passed his regrets on that. So never worry. Regan Mutimba is saying this is a super informative session, and Kennedy Kimani is also appreciating that this presentation is quite beneficial. And indeed, it's quite beneficial. So more questions. It seems there are no more questions on this. Now, Evelyn has just given some brief here, and I'll just read the way it is. Evelyn is saying, in the diagnosis of malignancies, flow cytometry is used as a confirmatory test after initial basic testing, including histology, cytology, and basic hemogram test. Highly recommended. Yes, thank you very much, Evelyn Webala, for these statements. I actually agree with them 100%. And thank you even for your participation. Phoebe is saying great presentation. No question from her. Thank you very much, Phoebe. And many others really congratulating, appreciating the presenter for this great presentation. So at this point, it's quite evident that this presentation is a super presentation, and it has been a wonderful one. And so also for flow cytometry in this era, is a very, very important tool in the field of immunology and also management and diagnosis of infectious diseases, and also just for characterizing various immunological parameters in the system to assist in um, management and also the overall health management among patients. And so all these underscore the benefit of flow cytometry. There are other areas that what has been presented today is just a minor arm of flow cytometry. Still, there are other areas that I think our presenter will still be onboarded to present on. So everyone is called upon to dig deeper into flow cytometry, the immunology aspect, and also um, accumulate knowledge in this area. So still very many appreciations are streaming in from the participants, and we say thank you. So at this moment, we are finalizing on this webinar, and I would like to bring to our attention the following. One,
Just,
for
just
yes, please.
George, just to answer my colleague, it was Kolo Ochieng on the dot plot discrimination in pediatric marrow. Yeah, I
Yes.
think if that was his question on hematogens. So they have the CD45 as a site scatter. So the lymphocytes, they are CD45 bright and then they are low site scatter. The hematogens themselves, they are CD45 intermediate low site scatter, but from continuum. And then plus, that is the ones that he was saying how to discriminate. They are CD45 dim. They have a low site scatter and often form a distinct tight cluster. So I think it's something straightforward. It's not something very difficult to discriminate the two. Thank you.
Wonderful. Thank you very much for that. And I hope Ochieng has got the answer. Duncan Awino, Mr. Duncan Awino, a lecturer at Kenya Coast National Polytechnic, is saying, pass my regards to Eric and is really appreciating your presentation. He's saying that is a clear and engaging overview of flow cytometry. Thank you very much, Mr. Duncan Awino, for your appreciative comment on this presentation. Yes, so as I said earlier, that I would like to bring to our attention the following one. For this webinar is ChemetTB accredited, and it will attract 3 CPD points. For one to be eligible for the points, then one has to pay and also attend a minimum of 60% of the duration of this webinar, so that they'll be awarded the 3 CPD points. In addition, the CPD points are uploaded automatically after the analysis has been done, and so we will report on your behalf the points once you are deemed qualified or eligible for the award. So this is done within 24 hours, and we do our communications on our various WhatsApp groups. So be very keen when we send you an email, just open and read it, because there's some of us who, by the virtue of the nature of work that we engage in, might confuse some of the figures in their ID or registration number. And so if any error occur in such a manner, then the CPD won't be uploaded as required. So we channel our communications on email, ensure that you read and respond as guided so that we can be in a position to assist and also serve you accordingly. We still have more forthcoming webinars, just be on the alert and we will deliver as you require. So at this juncture, I'll give our presenter just to give us a parting shot, just a 1 minute parting shot concerning the presentation and also any comment that he can have, or rather an overview or anything that you'd like to tell us, just a parting shot as we come to a close of this webinar. So, Mr. Eric, if yeah, you can hear me, you can just give us a parting shot, just a few statements that can summarize your work or can give a future course or rather anything that you can tell us and us as the audience. So, welcome, Mr. Eric, for this 1 minute parting shot. Thank you.
Yeah, thank you, George. Thank you so much, George. Thank you, and thank you everyone for attending this presentation. On Sunday, I went for a prayer day. So I was with Dr. Bishop to missing a children who encourage everyone to soar high. So the same message that he passed to all of us is, as lab fellows, you soar high. Do not limit yourself. The sky is always the limit. Do the best that you're doing. The little things that you do in the lab, do them to the best of your ability. You might never be rewarded where you are working, but there's somebody above who will reward you. Thank you.
Yes, thank you very much. Once again, on behalf of LMSK, we are very grateful and we appreciate your participation. We appreciate you, our presenter, Mr. Eric, and everyone who has managed to take part in this webinar. Once again, and finally, I would like to bring to our attention that LMSK has created a learning hub and we'll be circulating. We have been circulating the link so that our members can register for future seamless participation and also uploading of their web of their CPD points on the KMLTB portal accordingly. And so we will be launching and starting to use it exclusively as soon as possible. Updates on our WhatsApp groups. So stay put and we focus on how we can align everything to better this service. So at this point, once again, on behalf of LMSK, we say thank you and see you on our next webinar. So we come to a close of this. Thank you very much, everyone.
Thank you, sir.